It's a pleasure to be talking with you all. And I want to start with a brief territorial acknowledgement. I'm joining you from the traditional ancestral and unceded territory of the Musqueam community. This is the community that lived here for 10,000 years before European settlers came, and this is where UBC Vancouver is located. This is where I research and teach, as well as where my family lives. So I want to recognize this with an open heart and deep respect for these peoples. And this is my favorite place on UBC campus. This is the Rose Garden. And the question that I want to address in this talk is, how does the developing brain use its experiences to develop? And I approach this question largely through the lens of perception, or at least that's where I started. Now, why would you tackle this question through perception? Well, perception, and I'm using some visual examples here, is an ability that shows an incredible change in the first few years of life. You can see from the top here that babies are born with very poor visual spatial acuity, which changes dramatically over the first 6 months. Now, this is a representation of the sort of acuity or the fidelity of the information that our brains get from our eyes, but we also have incredible changes in the representations of these systems all the way through every level of the visual system. So for example, on the bottom here, you see pictures of humans and pictures of non-human primates. And what we know is that at 6 months, babies represent all of these types of faces somewhat equally, but by 9 months, 10 months, 12 months, babies preferentially represent and have much better, more specialized processing for the human faces compared to the non-human primate faces. They even subspecialize to the specific races that they experience if they're raised in a monocultural or monoracial environment. And this also happens with language, with speech sounds. So we know that the perceptual systems in babies change dramatically, and they specialize to their experience a lot over this period of time, over the first year of life and a bit beyond. And when I came to this work, there were two major theories about how perceptual development happened. One was bottom-up experience, which mostly said that what matters is the information that your eyes get. And maybe attention matters a bit too. Maybe what you attend to and you process a little more deeply matters, but probably mostly the thing that matters is literally what your perceptual system experiences, the input that it gets. And the other thing that matters is maturational constraints in critical periods, which is largely sort of an argument for genetic-based constraints and windows that would open and close for plasticity. Now, I'm not going to argue between either of these. What I'm going to suggest is that there's actually probably many ways that experience shapes the developing brain and shapes developing perceptual systems. And I'm going to focus on one other way that I've spent a lot of time investigating, and that is whether or not perceptual experience can engage higher level cognitive abilities and result in some top-down feedback that can really rapidly change perceptual systems. And this comes from the world of predictive coding. So these are some really classic references from this field. This comes from both vision science and cognitive neuroscience. And the idea behind this theory is that our cortex is very sensitive, not only to the input, but to what we predict the input to be. And here are some of the major models that came out, particularly in the first decade or so of work in this area. And what you can see from these models is that both of them have input or sensory input on the left. And this input goes through multiple layers of processing. And at each of these layers of processing, there's an interrelationship between feedback or a prediction of what that input should be and a feed-forward error signal. Now, I'm not going to argue that predictive coding is the only way to think of the cortex or even the right way to think of the cortex. But I think what's really ingenious about this theoretical approach is that it takes higher level information. And in this top model, you see it most clearly where you have the prefrontal cortex representing prior information and is this sort of higher level system. And it's feeding back to impact and affect activity at lower level systems. And specifically, it's predicting the upcoming sensory input. So my work started by asking, is this something that the developmental, or the work that I'm presenting today, is the developmental brain capable of something like this? This is very complex for an infant brain to do. Are infant brains doing something like this, having higher level systems interacting with lower level systems? And do we think that would matter for development? So to try to start to answer this question from this perspective, we'll ask, does sensory prediction modulate perceptual cortices in infancy? How are these processes affected in infants at risk due to premature birth? Where do these top-down signals originate? And what are their computational properties? And I'll end with a little bit of very brief future directions about where we think this work is taking us. So let me start with trying to answer this first question. Does prediction modulate perceptual cortices? And this is work with 6-month-olds by and large. And that's more or less, that's true for most of the studies I'm going to present today. All indicate when the age has shifted. But I did my PhD at Cornell, and one of the major themes at Cornell is this idea of ecology and thinking about the organism in its world. So I like to support particularly my experimental paradigms in sort of baby ecology. So here's my ecological example here. So imagine this baby is playing or doing something, and it hears a voice of its caregiver behind it. And what we're going to ask is whether or not their visual system has a prediction that they're going to see their father, has a prediction they're going to see their dad. And that's really what we're having in mind here, that the baby has some knowledge about, say, how sounds and visual events go together, in this case, the voice of their father and what their father looks like. And that specifically when they hear that sound, if they were about to turn around, their visual system literally is preparing to get an input of their father. Can we see that kind of prediction modulating their visual systems? And how we operationalized it in the lab looked like this for this particular study. So we didn't use voices and faces of their parents, but we taught them a new audiovisual association with a sound. It kind of sounds like, and it predicts a smiley face popping up on the screen. And what you see on the top right is they hear that sound and they see that smiley face 80% of the time after a period of familiarization. And then 20% of the time they hear the sound and we don't play the visual event. And what's crucial here is that for the visual omissions, as I'll call them, if the infant brain is responsive to the prediction of a visual event, what we see here is a large violation of a prediction of a visual event. They're expecting a red smiley face to pop up on the screen in this kind of dramatic way. And instead, they see nothing. So there's a big mismatch between prediction and input. And based on these models of predictive coding, we should expect to find a large response in their visual system. But if the infant brain is really just responsive to the bottom-up or sort of the sensory input that it gets, independent of its predictions, we should expect no activity because there's nothing that they're seeing. There's no visual input at all, or no change in visual input. So we did fNIRS recordings. These are relatively early fNIRS recordings. You'll see, and you have seen pictures of our more modern systems. Here we only had between 10 and 12 channels that really had good contact on the baby's scalp. But at this time, one of the things we did was we were among the first labs to do photo-based MR co-registration, where we took photos and measurements of the baby's heads for every baby who participated in the study, specifically as the sensors were on their heads. And we co-registered them to MR templates that were age and head size appropriate. And at the time, this was really considered an advance in terms of being able to localize where sensors were across the population and in the cortex, according to these average models. So this allowed us to create occipital and temporal regions of interest that were defined neuroanatomically based on our population, and we can see a representation in the cortex of where we were recording. And I'll show you the results. So we have the two regions of interest here on the right, temporal and occipital. The vertical axis here is oxygenated hemoglobin, and that's averaged in a time window that we decided a priori where we should see a strong response of the bold response, essentially, after the stimulus. So we picked that time window once and we never changed it, and we're averaging over that period of time. And more activity, more oxygenated hemoglobin is considered a proxy for more neural activity. And we'll start on the left here with the sound and the visual event. And what you see is both strong temporal and strong occipital activation, as you should expect. So they're both seeing something and they're hearing something, and their temporal and occipital regions of interest are proxies for our auditory and our visual systems, which is great. So moving on to what I'm calling the unexpected visual emission, because I've thrown in a control condition here. So they're hearing a sound and they're not seeing a visual event. So we still see a temporal lobe activation, as we should, because they're hearing a sound. But crucially, we also see a very, very strong occipital lobe activation. So they're hearing the sound, they're not seeing the visual event, and yet we see as strong of a response as when they actually see the visual event, suggesting that it's not simply the input that babies are responsive to, but actually something more, something having to do more with their expectations or their predictions. But we ran a crucial control condition because there could be potentially a lot of other factors that could be having an impact here, including the fact that there's this change from a baseline stimulus, which is lower luminance. And it could also be perhaps the babies just respond with their occipital lobe to any stimulus. Maybe they have some kind of indiscriminate responses. So I'll show you now results from a separate control study, a separate group of infants who had a similar exposure to the sounds and the visual events, but they were never paired. So when they hear the sound, they don't expect the visual event. They've never seen it together, and they saw the visual event separately. So this allows us to present the same stimuli, but now without any kind of expectation or any kind of prediction. And what you see is what you would expect, which is that you have a temporal lobe response to the sound, and you see no occipital lobe response in that particular case. So what's crucial here is that when babies expect the visual event to occur and it doesn't, you see a very strong response in their occipital lobe. But crucially, that changes. So these last two sets of bars here are showing you how quickly the system changes. When you take a baby and just bring them into the lab and play them the sound, their occipital lobe does not respond. But with just a couple of moments of experience of learning that sounds and visual events go together, all of a sudden the profile of response of this region of the brain changes quite dramatically. So this really had a big effect on me as a researcher. This was really a profound result for me, that the infant brain was able to change very quickly, particularly its perceptual systems were able to change very quickly, and they were able to change very quickly in this predictive way. So we followed this up in many ways to make sure this is not unique to this study or to this population. One way that we followed this up recently is by doing a study with neonates. So these are babies who were born within the last 48 hours. So they certainly had visual experience, but neurally their brains are much less mature. And we thought that at this point, certainly babies would not be able to do something as sophisticated as this. And we picked a very similar paradigm. We made the learning context a little more simple and the stimuli more simple, especially since babies were sleeping during the study, but we found qualitatively the same effect. So this is work that was still in progress. So I'm just going to present it to you briefly, but I'm happy to talk about more details in the Q&A at the end. But going from the top here, this is the response in the occipital lobe when babies hear a sound, so we're representing that as probability of an auditory event positive, and when they see a visual event. So this is like the 80% of the time that they see them together, and you see a response. The next one is where they hear the sound, and now they don't see the visual event. The visual event is violated, and you see a very strong response. And the last is for the control condition with a separate group of babies that are now hearing the sound without the visual event. So these are the same three groups, now focusing just on the occipital response. And I'll show those now in a bar graph that's very similar, where again, we picked an a priori window. You can see that shaded in the temporal plots, and you can see we see very strong response in the violation, and that's very different from these control participants. So even neonates are showing this incredible rapid change based on this experience and based on their learning and predictions. We've explored this in different paradigms. So here's a paradigm about sequence learning that's inspired by this amazing study, Bazarat et al. 2014. And in this paradigm, babies see sequences of 4 shapes, and one of them is an oddball, one of them is unusual. And for folks who are familiar with oddball or MMM type paradigms, they're usually done in EEG. And this is our attempt to modulate that for fNIRS. So we have these blocks, and the blocks are either when at the top where the oddball's location is unpredictable. You do not know where the oddball is going to come up. And we're looking at the neural response using NIRS for the entire block. And we're going to contrast this with a situation where the oddball is predictable, where you always know where the oddball is going to occur. And these again are in 6-month-old infants. And what we wanted to know is whether or not they could reduce their sensory response to the oddball. This is qualitatively a similar finding linked with predictive coding as well. And this idea that higher level learning can impact perceptual responses, but here it's in a sequence context. And here what you do is you expect a reduced response to something that could be a violation, could be an oddball, because you're able to predict it. So it's looking at hypothetically or theoretically the same thing, but in the opposite direction. And what we found, oh, I forgot to mention a crucial detail. Each of these shapes also has a unique sound. So it's audio visual. And that's because we wanted to see, was this going to occur in the occipital lobe, and was it going to occur in the temporal lobe? And it could be both. We were also interested, and this was our first direction, in seeing whether or not the frontal lobe might be involved here. Because the EEG results from Bazarat et al. Suggested that the frontal lobe of babies might be involved in this kind of learning and detecting these kinds of changes in the sequences and the structures. But they didn't look at what was happening in the perceptual systems. So here are the results. You can see the localization. Now this is a different fNIRS system that has 70 channels as opposed to 12. So that was a big step up for us. So we can look at many more regions now. And I'm going to show you predictable, unpredictable in those 2 colors, purple and yellow, and then the contrast between predictable and unpredictable. And that's going to be when in our time course, we see a difference between the 2 conditions. And that's corrected, strongly corrected for multiple comparisons, because we tested it every time point. And I'll show you the results from the occipital and the temporal response. And here what we see is that you find a stronger response for the unpredictable compared to the predictable, which is what we expected. So in the predictable case, the babies are able to reduce their response to the oddball design, to the oddball that shows up, and overall show less sensory responses, likely because they're using their prediction to do so. And we found, this doesn't tell us that the frontal lobe is what is causing this effect for sure, but we did find that the frontal lobe was involved and in the same direction. We actually were ambivalent or unsure which direction the frontal lobe would go here, but this is the direction it follows with the sensory responses. So just before we go back to sort of the answer for this question for this part of the talk, I want to highlight one key aspect of methodology for NIRS. So the first study I presented showed this picture-based, used this picture-based MR co-registration. But the second study is actually using some new technology, which is we think is really helpful. And I want to highlight what's useful about this and where I think the field is going. So let me see if I could, oh, no. I was hoping I'd be able to get this link. I can always send this afterwards, but so what this is showing here is that what we're doing now, instead of this rather laborious and manually coded MR co-registration approach that we used before based on photos, now we're using cameras and videos to use a combination of machine learning and computer vision to automatically build a head model of the sensors on the head. And we then did all our validation work on this in adults, but what the link would show you is that this also works in babies. We can take these videos very easily in babies and do this co-registration process. And in fact, in adults, if you compare a 3D digitizer, which is quite an expensive piece of equipment, the Pohemus, the top shows you sort of test retest on the same locations done twice. You can see that the error is decently large, right? It's about 5 millimeters. That's what the histogram at the top shows you. But when you take a video twice, you actually see that our error is better than the 3D digitizer that costs many thousands of dollars. So you don't need to have expensive equipment to do something like localize sensors. And this largely you can use out of the box computer vision techniques to do that. So if anyone's interested in this technique and we're using this type of method, please reach out to me. We're very happy. We're trying to develop some open source toolboxes and share these methods for other labs to use. And this type of co-registration is very important for fNIRS because, of course, we're recording from the scalp, but we ultimately want to be saying something about the cortex. But there's a number of things working against us there. But I'm going to tell you the good, the bad, and the ugly of this, making a joke on the famous American movie. The good is that using this method is quite precise. And in fact, we can localize fairly specific regions in the brain using this. So this is an example of us trying to localize the LOC in babies. And I think we did that quite well. And we found shape selectivity of this region at 6 months using this technique. And that's outlined in this particular article here. The bad and the ugly to some extent is that when you look at where these channels are using these localization techniques, they actually cover a very large swath of the cortex overall. And while there are some regions we can really localize somewhat specifically, the LOC is quite large, really, when you talk about like a cortical area, especially a posterior one. And so by and large, I think we're kind of stuck right now with these big regions of interest. And that sort of highlights or foreshadows some of the work I'm going to present to you now, where we're, for example, looking at the entire frontal lobe. I think we can subdivide a little more, but I think it's hard to do so given the realities of what our recordings look like. So you can see in this diagram, if you take one channel and I show you where it's presented on all of the infants in the population with the scatter plot on the bottom left, and then I show you how big that channel actually is on the size of a baby's head and their brain, which is quite small, it's a big part of the back of the head, right? It's a big part of the occipital lobe, even though it's just a single channel. So we're recording very large regions. But I think the good thing and the direction that we're going, we recently got some funding from the Canadian Foundation of Innovation to move towards more high density recordings with infants. So this is one of the systems that's available now, the Gower Labs Lumo system. And it uses these tiles that have many sources that are embedded in them. And they will hopefully allow us to move towards a more high density tile of recording. And with that, I'm hopeful that we're going to get closer to, not to fMRI, of course, but have more density of recordings that will allow us to do something more sophisticated and get smaller and more refined regions of interest. So what you can see here is what a typical fNIRS system now looks like, and all the studies I'm going to present today use that on the left, and in the middle shows you a schematic of what the high density system would look like. And then you can see an example of it on the right, which is also essentially wireless, which is very cool. So going back to our overview, Does sensory prediction modulate perceptual cortices in infancy? Our answer is yes. We looked at 2 different tasks, and we've looked at 6-month-olds as well as neonates. We've done a couple of other related studies, but by and large, we're finding good evidence that babies are modulating their sensory systems, their perceptual systems to prediction. And they do it rapidly and quite intelligently. And this seems to be available to some extent, even very early on in life. So now I'm going to present to you our work in premature infants and how this process plays out in that population. And so our question is, does premature birth disrupt prediction-based changes in perceptual cortices? And I'm sure you all know premature birth is a risk factor that puts babies at risk for a number of potential medical and developmental difficulties, including language delays, motor problems in motor abilities, learning disorders, autism, and reduced IQ, among others. And part of the reason why that might be is because the brain is changing so incredibly over gestation. So what you see here is what the brain looks like in human infants at different weeks of gestation, where 40 weeks is considered, well, 39 to 41 is considered full term. And in this particular study, we looked at babies born 32 weeks and under. So we're looking at quite an extreme population, relatively extreme population in prematurity. There's a lot of babies born in a moderate preterm range, but we're looking at the very and the extremely preterm infants. And these are our samples. We have our full term group, which are 6-month-old infants, and we have 50 of those. And we're going to contrast them with a preterm group. There's also 50. Those were tested at 6 months corrected age. And crucially, they have no known predictors of cognitive delay. So these are beyond being premature. So they had very benign medical courses. So no IV, IVH, but no interventricular hemorrhage, no PPL. By and large, they were really the most healthy preterm babies we could get. And this is work I did with one of my wonderful full-time research staff members, Alex Boulding. And this, incidentally, is also using the same paradigm that we started with, where babies hear the sound and they see the visual event. And they see that 80% of the time, and then 20% of the time, we play the sound without the visual event. So that's the same paradigm. We just have a preterm group now added. And we also have the same 2 regions of interest. On the left, you can see the temporal lobe, and on the right, you can see the occipital lobe. And this is the same data that I presented before, but now in a different configuration. So on the left is all the temporal lobe, the right is the occipital lobe. We have things coded for visual present and visual omission. So on the left, what you see is that you get strong temporal responses in the full-term babies when they hear the sounds, regardless of if they see the visual event. And on the right, you see a strong occipital lobe response in these full-term babies when they see the visual event in the light orange, and when that visual event is violated in the darker orange. So this is a replication in the first study. But how does this look in the preterm babies? Well, with the temporal region of interest, what we see is we do see a response in the temporal lobe to the sounds. And in the occipital lobe, we see a response when they see the visual event. It is attenuated from what we saw in the full-term babies. And I'll get back to that in a second. But crucially, what do you see when the visual event is violated? What we thought, and especially given that we saw this attenuated response in the visual present, was that it's possible the response is attenuated in preterm babies to the visual event. But if they're responding with these types of prediction mechanisms, if you're getting this prediction-based changes in their responses, we would expect that response when the visual event wasn't presented to be similar. But if the response is not present, we would expect that response, if the prediction was not modulating their perceptual systems, we'd expect that response to be much lower. And that is in fact what we saw. We saw that there's a significant difference in the visual system or the occipital lobe of preterm infants when they saw the visual event and when the visual event that they expected was missing. And there's a significant interaction of trial type and birth status in the occipital lobe specifically. We found no differences in the temporal lobe at all. And what this suggested to us was that, well, first, there's a fair amount going on here, and we'll actually look at this data in a different way later, thinking more about what's happening over time in this learning task. But what we see is that it doesn't seem like prediction is modulating the visual system or the occipital lobe of preterm babies in the same way that it is with full-term infants. And in fact, preterm infants don't even look like they're responding identically to the visual present event. One of the things we looked at here was whether or not there are interactions with gestational age and how socioeconomic status plays in this population. So taking a look at simply the response to the violation case, where the visual event is violated, how strongly does their occipital lobe respond in that case? We can take a look at that response over the gestational age of the babies in the task. So that's what we have over the horizontal axis here, from 23 weeks, which is the youngest baby, to full term. Remember, all these babies are tested at 6 months or 6 months corrected. This is just when they were born. And what you see is that there's, of course, variation. But by and large, we didn't find any difference across the range of gestational ages in our preterm group. The babies who were 30 to 32 weeks gestation were just as impaired or had just as much of an attenuated response as the babies who were born quite a bit younger. So it doesn't seem that within even this quite large period of prematurity that we're finding any modulation, which is interesting, and it suggests that perhaps moderate preterm period is really crucial in the development of these abilities. We also took a look at socioeconomic status here. We have the babies again coded by the two different groups. And what you'll see is we have a much larger spread of socioeconomic status in preterm infants compared to full-term infants. By and large, our full-term infants are middle class or upper class, and preterm babies are actually by and large upper class and lower class. And while we did find an effect of socioeconomic status, if you control for socioeconomic status, it doesn't explain the prematurity effect. And we looked at that a couple of different ways. So SES is certainly involved here, even at 6 months, corrected age, but it's not explaining this effect, even though there's these differences between the populations. One thing we didn't worry quite a bit about was whether or not the reason why we don't see this effect is because the babies didn't learn the audiovisual association. There's some suggestion that premature infants do struggle with audiovisual learning, so we wanted to make sure that they were learning in this task. We thought that they were because the learning component of this task is extremely easy, and the tasks in which there are differences in audiovisual learning in premature infants are quite challenging about the learning particularly. So we actually ran a whole separate control study with another 100 babies, where this is a purely visual study, or a behavioral study. We didn't do a NIRS component here. But what we did was we gave them a familiarization period to the audio-visual sounds. And then we gave them a choice, whether or not how long they wanted to look at the sounds and the visual events repeating together, or where there were 50% omissions. And the idea here is that if they learned that the sounds and the visual events go together, they should show differential learning, differential responses to these two different trial types. And what you saw across the birth status, we have full-term babies on the left and preterm babies on the right, is that you see that both groups show differential responses. They prefer to look at the audio-visual coming together. So we would call this a familiarity response. And if anything, the preterm babies are better at it. So if anything, they're better learners. If you do a direct statistical comparison, there's no difference between the groups. So we don't think the reason why neurally the preterm babies don't show this prediction effect is because they're not learning the association. And I'm actually going to show you additional evidence to that in a moment. But they are learning that these two things go together, but they're not able to use that knowledge to feed back, to change their neural responses in their perceptual systems, in their visual systems specifically. So the answer to question number 2, how are these processes affected in infants at risk due to premature birth? I shouldn't answer yes to how. The answer, are they? Yes. We still don't quite know how, but they're not arising from task differences. That's the crucial thing. So we'll talk a little bit more about premature infants, and while we also talk about where do these signals come from and what computational properties do we think these signals are carrying. And in order to discuss that, we'll discuss, let's go back to one of the predictive coding models, just to remind you from the beginning. So like I mentioned, I think this one in particular is a nice one to think of for the tasks that we're using, where on the left, you see there's a sensory input that goes up across many different levels. And at the highest level, what you see is there's this encoding of prior information. And here that's represented in the frontal lobe, in the prefrontal cortex. And that information, that higher level information, feeds back to many different earlier systems to modulate activity. And there's this interplay between feed forward and feedback dynamically that allows the brain to respond predictively, to both learn about the environment and respond in its sensory processing as well. So this was our working model, and we're instantiating it in young babies in this way, where we have perceptual systems on one hand, and we have cognitive abilities and systems that learn about environmental structure on the other. And this is a 6-month-old baby here. And what we've generally been arguing is that in full-term populations, what we find is that there's this feedback from cognitive abilities and systems that learn about environmental structure, and they affect perceptual systems. And we've been calling that top-down sensory prediction. We've been arguing that full-term babies have top-down sensory prediction and pretty much from birth, and that this seems to be impaired when you're born very and extremely prematurely. But where do these signals come from? I've kind of said, cognitive systems and systems that learn about environmental structure very broadly. I think they could potentially come from many different sources, but we see time and again that the frontal lobe might be involved. So we wanted to look at that more specifically. So the question, is the frontal cortex involved in top-down sensory prediction in infancy? And there's a fairly well, I don't want to say long tradition, but there's been a number of studies now that have been interested in what the frontal lobe is doing in even very young babies. And that it looks like the frontal lobe is responding quite a bit to tasks, like learning tasks, but particularly to violations in what the babies have just recently seen and heard. So here's an example. These are from 3 and 4 month month-old babies sleeping in a habituation type task where they hear blocks after blocks of ba, 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 the same syllable over and over again. And this is the response in the prefrontal cortex, the channel in the prefrontal cortex, and you can see it responds a lot at first, and then it really responds much less, and that's in the red. The blue is considered a control condition. And then the blue is the deoxygenated, the red is the oxygenated, I apologize. And then for a period of time, they switch. They present babies pa, pa, pa instead. And what you see here is a very large response, again, of the prefrontal cortex when there's a change from ba, ba, ba to pa, pa, pa. Incidentally, you don't see any difference in the temporal lobe or regions that are responsive to auditory processing on their own. In babies of this age, the only differential response that the researchers saw was the prefrontal cortex. There's also some theoretical views that point to the prefrontal cortex here as well. So there is a review of sort of the developing brain and prediction more generally by Gisela DeHaan-Lamberts and Liz Spelke in 2015. And they mentioned the first study I presented, but they specifically said, we wish you had recorded the frontal lobes, the frontal areas. They specifically would want the frontal areas to be responsive, to be recorded to test for hierarchical models and the role of top-down modulation in early learning. So theoretically as well, folks think that the frontal lobe should be involved in these types of tasks. So this was actually a modeling work, a modeling paper that we did on the data from the premature study I just showed you that had the 100 babies overall, 50 preterm, 50 full term. We took that data and we used some computational learning models to try to understand the trial by trial variation of those neural responses, and specifically what is the frontal lobe involved and what kind of information the frontal lobe might be communicating to the occipital lobe here. So to do that, I want to highlight that we've been really talking about this task as being, there's the audiovisual trials and then these visual emission trials. There's just two types. But really in the task, there's many, because babies they don't know how we've designed the tasks. They're just watching these stimuli over time. And it could be that there's 3 audiovisual trials in a row, as you see on the left here. That means there's very low error when babies see that smiley face. Or it could be on the right, where they see 2 omissions in a row and then they see the smiley face. That would be a very high error. And so there's this trial by trial change in the amount of error, and that's what the delta is here, the amount of error that babies might experience on any given trial. And that's related to whether or not it's an omission trial or an audiovisual trial, but it's also beyond that as well, right? There's variations in error in both types of trials. So we exploited this using the Rescolar-Wagner model, which simply says it's very similar to predictive coding in the sense that you take your probability of a particular event, you observe what the visual event actually is, and you update your internal expectations, your internal probabilities of getting an event, and you calculate error. So that's what that delta is. And so what we did was we took 2 of the regressors from this model. We fit this model on the task. So this is model follows what the task should be. And based on the regress, or based on these certain parameters of the model, we use that as regressors in the neural data to see what regions of the brain follow closely and how closely how closely do they follow these computational components of the model. And the 2 that we modeled were error squared, delta squared, which is simply how much error. And the reason why we do the squared is because we don't care which direction, a positive error or negative error, but that's kind of a detail that's not important. The other thing, so that's in the box on the right. The box on the left, the other thing that we looked at was whether or not there was a visual event. It showed up or it didn't. And that's very similar to our original finding, where you say you're contrasting the 2 trial types. So this is what we see. On the top is the stimulus regressor. And now we have the 2 colors confusingly being full terms and preterms, and our regions of interest along the horizontal axis. And what you see here is that we get responses when the differential responses when the visual event shows up versus when it doesn't. And that is to be expected, right? And incidentally, you see it strongest in the occipital lobe, but you sort of see it everywhere. And this is sort of one of the things that we know, is that we need these contrasts to be able to show more specific effects. If you just simply look at what parts of the brain respond to a stimulus, in general, the brain is responding to new stimuli. But crucially, for the prediction error, where do we see? Well, in the frontal lobe, we actually see very strong responses to the prediction error. So the frontal lobe is modulating its response depending on how much prediction error there is, with greater response coming from greater error. And that isn't different between the preterm and the full-term population. This is confirmatory of our behavioral findings that in fact the preterm babies really are responding quite strongly to the learning context. They're learning what's happening here. They're following the task. It's not that they're not learning. But when you look at the other two regions of interest, now is when you see sort of differential responses between the preterms and the full terms. The full terms show responses to these errors in the occipital lobe, and they incidentally do in the temporal lobe too. But the preterm babies do not. They are not following, their occipital lobe doesn't change its response with the error. And again, this is very synergistic with the original finding, but it just approaches it from a very different perspective, a very data-driven perspective, constrained by these computational learning models. And the way we interpret this, because we don't expect the occipital lobe to be doing these error calculations on its own, that's not really the role that we think the visual system plays. We think that in the full-term babies, the occipital lobe is getting that information from the frontal lobe, that there's some connectivity there. We can't directly see it, but that's our inference. And that connection is not present in the occipital lobe. That the frontal lobe in preterm babies, that's not present in preterm babies, is what I meant to say. The frontal lobe is tracking the error over time, but it's not communicating that error back to the occipital lobe. I will make one note about what the temporal lobe is doing here. In this particular model that we ran, there is a difference between the temporal lobe between the two groups of babies. We ran 3 or 4 different versions of this learning model based on some sort of a priori determined variations in how the field, computational reinforcement learning field, thinks about these models in terms of the flexibility of the parameters, for example. We made more simple models and less simple models to see where did we find convergence. And this happens to be the only model that shows this difference between the temporal lobe. I show it because it's one of the easiest to explain and the most standard in the field, but we don't really make a strong interpretation from the temporal lobe because it doesn't show up in all of the models. All the other effects are consistent across every version of the model that we ran. I think actually I'm going to skip over this part just a bit. Let me think. I'll present it. This is very, very similar. This is similarly taking the same data and looking at it in a slightly different way. It shows the same effect. So again, we've been looking at these audiovisual events and these visual omission events. But one of the things I didn't mention in detail is that to get these effects, we give babies this period of familiarization where we present a bunch of audiovisual events all together. So we're calling these learning blocks, and we present a whole lot at the beginning, and then we intersperse them through the trial in order to keep their strong expectations that the sound and the visual event go together. And they occur throughout the study. They occur at a high density at the beginning. And one of the things that we've done is we've taken a look at those learning blocks and the responses to those learning blocks over the experiment to see which regions are changing their profile of responses over time. So we think of this more about tracking learning because there's no difference in the stimuli. It's just how often the baby has experienced it. And we're looking at it in this region of the frontal lobe, this region of the occipital lobe, and this region of the temporal lobe. And looking at the data from the very first experiment here, what you see is that there's this rise and this fall of activity across all 3 regions of interest. There's a significant rise in all of them, and there's either a plateau or a taper, depending on which one you look at. And this is very consistent with what a lot of people think of with baby learning, is that there's sort of increase in familiarity, and then there's sort of a decrease over time. Looking at the big full-term preterm cohort that we did, in that one, we see the same thing in the full terms, where we see increases and decreases over time. And you can see these are the model fits on the right. The linear shows is there just an increase, and the square shows is there a nonlinear result here. And on the left, on these plots, you see all the individual babies plotted, so you can see the variation. And in the bolded line, you see the average. Here's what we see in the preterms. In the preterms, we see significant changes. We see significant modulation over time in both the frontal and the temporal responses. And we do not see any modulation with time at all in the occipital lobe. And again, this is another way of saying the same thing. These preterm babies are not changing their responses. I'll show you in contrast across the 2 populations. They're in the occipital lobes particularly, which is the target of the prediction in this task, where there's variation, there's changes to what can be predicted. we do not see that these babies are changing their responses. It's just flat. But their frontal lobe is really doing what it should do. So it seems like there's this disconnection in this brain, right? Where this information is not making it back to the occipital lobe. When we look at which responses here and which trajectories predict how strongly they respond to this violation, so the violation was really our measure of prediction here. Oops, I didn't show the response, but that's the occipital lobe. The responses in the occipital lobe are the only one that predicts how strongly they respond to the violation. So that suggests that how much you modulate that occipital lobe while you're learning really predicts how strongly you're responding to violations of your prediction or these sort of changes based on predictive processing. Now, this has been a lot of data. This is the very last one I'm going to show you. And this is taking us to the direction that we're going now, which is thinking about connectivity in this picture. So I've shown you all these sort of suggestive findings that the frontal lobe is involved, and we think the frontal lobe is sending that information to the occipital lobe, but we're really not measuring that directly. In part because with baby NIRS, measuring connectivity during tasks is a challenge. But this is our first attempt here at doing task-based connectivity. And it's in the task that I showed you, one of the tasks I showed you earlier on, which was the sequence task, where babies see 4 shapes in a row, and there's an oddball stimulus that appears, and it can either appear in an unpredictable location or a predictable location. And this started off in part as a replication of that effect with extending to the parietal lobe, as well as a connectivity study. And this is by Soribach, my current graduate student. And what we have here, the red is predictable, and the blue is unpredictable. And what we see is we find the same findings again now in a new group of babies, that we see greater responses for unpredictable and attenuated responses for predictable. And we see that in the occipital lobe, which is where we saw it before. We see it in the frontal lobe, which is where we saw it before, but we had not recorded in the parietal lobe, and we've recorded there now, and we see differential responses there as well. Sori is very interested in the higher level systems, the sort of dorsal attentional systems. And so we were zeroing in on the frontal parietal network here, and we were wondering if there was differential connectivity in this network across this task. And specifically, we hypothesized that there would, even though there's more activity in this univariate way during the unpredictable condition, we hypothesized that there would be greater connectivity during the predictable condition, because we think that connectivity is necessary for attenuating this response or changing the responses during learning about predictiveness. And that's essentially what we saw. So here are our different regions of interest combined here. We have frontal parietal connectivity, and we see decent connectivity in both conditions, but we see more connectivity in the predictable, that's the PS. We see decent frontal occipital connectivity, and there is more numerically during the predictable condition, but it doesn't reach statistical significance. And we don't see any difference at all in connectivity in the parietal occipital regions together. So what we've taken from this is that, first of all, this frontal parietal network is pretty active in these young babies. Again, these are 6-month-old babies. And it seems to be involved in this task on sequence learning. Specifically, we're finding greater frontal parietal connectivity during the predictable tasks when babies can learn something new and predict what's coming up next, as opposed to situations where babies can't predict at all and they're not really learning anything about the sequence. One of the puzzles for us is what's happening with the occipital lobe, because we were expecting to find connectivity with the occipital lobe that was modulated with this task, and we just didn't see it. And we don't know if this is a power issue or what's happening here, but this is something that we're continuing to work on, because of course we'd really like to be able to see that there's connectivity to the occipital lobe, since that's the region that we're really seeing its response modulated in these learning contexts. But I'll wrap up now with a future direction that's kind of taking us in a bit of a different place than we've been before. So along this big line of work, we've been really looking at the data and asking ourselves, what does it mean? And one finding that keeps coming up that is puzzling to us is that babies' brains, when we investigate them, look very connected and very dynamic. But here I'm showing you some really recent and really beautiful resting state data with babies that are actually quite a bit older than the babies that we study. These are showing 11 months to 36 months. And this again is looking at the frontal lobe and the parietal lobe as well as the temporal lobe. And what they find is there's not very much connectivity in this network. It's there at 11 months, barely, and it increases quite a bit. So when you look at these sorts of networks in resting state, it doesn't look like the baby brain is very connected. But we see the baby brain as being very, very connected when it's doing a task, when it's learning to predict things, and when it's learning something new. So, and I'll also say that going back to the premature babies, in the premature babies, we're seeing in our tasks that it looks like that brain is very disconnected, that prematurity results in sort of disconnection between the frontal lobe and the occipital lobe. And sure enough, connectivity is something that is a very consistent neurophenotype, or reduced connectivity is something that comes up a lot when looking at premature infants. So this is a nice example from Chris Smyser at WashU, where he's looking at neural connectivity, again at resting state, in term babies, preterm babies, and then contrasting them. And you see that overall, there's much more connectivity in term babies than preterm babies. And you can find these differences in connectivity in this population through to adolescence and beyond. So this is quite a stable phenotype for preterm infants. So they show less connections than term babies, but they also show less connections in these more cognitive tasks, these prediction tasks that we're doing. So one of the directions our work is going now is to ask, how does the engagement of large-scale networks, like during prediction and other cognitive tasks, how does it relate to the development of long-range neural connectivity? We think, and there's some theoretical reasons to believe, that how strongly the brain responds during tasks might lead the development of the anatomy, might lead sort of the coherence and maturation of anatomy that might show up in resting state. So one of the things we want to do is to look at the interrelationship between the engagement of the brain in a task, like the engagement of the brain during these prediction tasks, and the engagement of those same networks during rest, and to see how they interrelate across populations and over longitudinal time. So broadly, my work speaks to how does the developing brain use its experiences to develop. And we've been looking at prediction and whether or not that model is an applicable model here. And we really argue, yes, babies are much more sensitive to predictions neurally and are responding in a very sophisticated way to a situation where they can predict what they're going to see next. These mechanisms seem to be impaired in premature infants, and we think this could be significant for their development. And we think that for full-term babies in particular, that regions like the frontal lobe, but potentially other higher level regions as well, could be feeding back information to the occipital lobe, and what they might be tracking is prediction error. So I'll sum up quickly with a talk tweet. This is a technique I've been using just to try to have my summary be very short. And here's my tweet. The infant brain is sophisticated and dynamic when it is engaged in prediction. It's modifying perceptual cortices rapidly and in collaboration with the frontal lobe. How does this relate to the protracted development of long-range neural connectivity? And I'd like to thank the families who come in, my new lab, the UBC Baby Learning Lab, the amazing research staff and students that I get to work with, and our funding organizations. And thank you all. Merci beaucoup.
Much, Lauren. That's the camera, right? Okay. So thanks a lot. It was a super interesting talk, very elegant data, and an amazing story you just told us. So, est-ce que vous avez des questions dans l'audience? So we'll start with the audience for questions. Est-ce que quelqu'un a une question pour Lauren? Si les gens préfèrent la poser en français, c'est correct aussi. Puis on peut. Oui.
Well, I'm not sure I understood well how the 3D digitizer versus the video to video were comparable, and how much was it in the difference? Because you said the 3D digitizer was so much more expensive. And so is it just the error was the only difference, or were there other things?
Great question. One second, let me go back to that, that part. I'm so glad you asked about that. Yes, so I didn't really go through this in a lot of detail. We have a whole paper on this. And it's out now and available in open source as well, which I think is nice. We did a lot of different comparisons here, and this is really just, I think, one of the more easy to explain ones, but by and large, we see the same thing across all the population, all the comparisons we did. So the top is looking at the 3D digitizer to the 3D digitizer. So that's basically saying if you have one participant in a cap, and I go through with the digitizer and I get a recording of every location of the sensor, and then I have the same person in the cap and I just do it again. What's the error between those two? So this is like test, retest. How consistent is it? And this just gives you a sense of the amount of error of the system, of this measurement. And that's what you see on the top right. That's that histogram there. This is how much of a change in millimeters, this is based on Euclidean distance, between the first location and the second location for that same sensor using it's the same person and it's the same digitizer, you're just doing it twice. So it's like the noise. And what you can see, it's certainly not bad, but it's not that great either. Just the sensor itself is giving you an average of about 5 millimeters of error. And that's, yeah, that's, I mean, it's decent. What we found though, which was actually a surprise to us, part of the reason why we did this is we wanted to make sure our video technique wasn't poor in its localizing, and we fully expected to be comparable or slightly worse than the digitizer. We just wanted to know how much worse. But what was to our surprise is that when you did the same thing but with a video, so you have one person and you take a video and then you use our software to localize the sensors, and then you take another video and you use our software to localize the sensors, there you get an error too in Euclidean space, and we actually did better than the digitizer, that by a couple of millimeters, well, about 1 millimeter, but it was significant. So that's what we're trying to show here, is that the digitizer costs more, and it's in this particular measure, it's actually worse. We did better using videos and these analytic techniques than the digitizer that you spend several $1,000s for. I'm happy to talk more about this. Did that answer your question?
Well, yeah, I'm also interested in that software, and but I'll check my own.
Yeah, and please, if you're interested in the software, so we're still working on a toolbox for it, but I will say that in principle, none of it is very complicated. So from a computer science perspective, these questions of using a video to create a 3D model is a known problem, and there's actually off-the-shelf free software that you can use to do that. So we modified it to make it like kind of more like a toolbox for us, but these techniques are very, they're not, it's not like we came up with them. Computer science has, and we're just exploiting them here. So if you take a look at this reference, and please write me, anyone who's there who's interested, and I'll do my best to point you to the direction so you can do this too. Ultimately, we'd love to be able to either share what we've done with the field or point people to resources that are ideally freely available or very inexpensive to do this.
Thanks. Thank you, and thanks for asking that question, because I was wondering about your software too. So I'm gonna contact you too. But I guess that the fact that we don't have the human error with that kind of pencil with the polymus, So the fact that you're using the video and that the algorithm, that you don't have the human error, I guess it explains a bit of the better resolution you have.
I think so. I think that could be part of it. I mean, certainly I think human error is one of the reasons why the digitizers are not as precise as they could be. I think another reason is that those digitizers are extremely sensitive to other sources of metal and other magnetic sources. And this is in actually a very tightly controlled situation where this is one adult and we're doing this twice. So this should kind of be the optimal situation for that. But then you think about doing this with babies, they have a parent sitting on them. There's only so invasive you can be with the parent as well. Do you have them take off their jewelry? Do you have them remove their cell phone? Like they're moving around. So the digitizer actually probably performs, for those reasons, combination of human error of just trying to deal with the participant, as well as all these other sources of magnets and metal and everything else. Yeah, there's a lot of sources, and it's even worse in babies, I guess is what I'm trying to say.
Yeah. Any other questions?
I do. So Lauren, I would have liked to have you actually here. It's really, and have dinner with you tonight. And so we could discuss a lot. So actually we also just got a paper accepted where we did tweak a little bit the Becerra task in babies using EEG, and we choose to focus more on the auditory modality in terms of prediction and repetition effects.
Right.
You choose to focus really on the visual aspect, so it's really neat actually to see this. Was there any reason in the first place that you choose to focus on the visual?
Oh, thank you so much for that question. I'm so excited about that work too. There's so much incredible work in audition happening here. Why did I pick the visual? Well, going back to the very first study, which is the study that launched this line of work for me, I was working with Dr. Aslan at the time, and he, of course, does lots of audition work, but he's also, I think, in core a vision scientist. And I had just come from Cornell, where a lot of folks are folks who study language development and psycholinguists. So I was a little enthusiastic to start in vision. So that kind of was part of it historically. But also, I really wanted to look at it in terms of audio visual, particularly because I felt like from a connectivity perspective, that was really hard. I kind of wanted to create a task. My original plan was to sort of create a task that babies would fail at, to be honest. I really did not expect them to do as well in these tasks as they do. My plan originally was to do this audiovisual task to show that they could not do it, and then to increase in age until they could do it. That was my plan. And I think the audiovisual component, part of the reason why I think it's hard is because the connectivity that might be required there. So, I think that it's not that I think the unimodal case is trivial, because I really do not think that it is. And I think that you were likely in the unimodal case, either audition or vision, that in many cases, I think those systems will also rely on other systems for the learning and prediction part of it. But one sort of for very, very simple types of MMN type stuff, until that work really got pushed into the predictive part of it, there's some folks who thought that, if it's just ba versus pa, that's something that the auditory system is doing on its own. So I kind of wanted to stay away from that, because I didn't want to have it be so simple that you wouldn't really be relying on these other systems. Now, like I said, I don't think that's what's happening in the auditory case. I think it's very easy in the auditory case to start to employ the frontal lobe and other regions to modulate responses. But that was one of the reasons why I wanted to make it harder, and I wanted to make sure that other systems would have to be involved to modulate the occipital lobe in particular. But may I say one more thing, which is, this isn't something that's published, but it's something that we've done some work on, which is, let me go back to that slide again, 1 second. With the original task, this one here, we've tried to flip it around and make it an auditory task where the vision predicts the auditory. And we have really struggled with that. This is, I think part of it is an uninteresting, I think on one hand, there's kind of an uninteresting reason to it. When we flip these stimuli around, so the visual event showed up and say like hits the top of the box and makes a sound and then comes back down, the babies just hated it. They cried and fussed out of the study at such an incredible rate. And it was so bizarre to me, confusing to me, because they loved this version of the study. In fact, this was one of the best study I've ever had in terms of the baby compliance. So it's so confusing how you can just take the same stimuli and switch them and the babies hate them. So yeah, but we've done some other tasks where we've tried to do it where the visual predicts the auditory. And we found that very tricky, where even just for ourselves, it's hard to think that one really produces the other. Oftentimes it feels more like they come together, more like an audio-visual, like a toy that like you shake it and produces a sound, as opposed to one is kind of leading to the other. It feels a little more like predictive or causal. The other thing is that when we do have a TAP paradigm that sort of works, we see that the corresponding auditory effects kind of fall in the middle. They don't look like as strong as the visual ones, but they also don't look like there's not predictive responses at all. So we've wanted to flip it around and at least make this cross-modal paradigm an audio-focused one, and we really haven't been able to do that. We have some work that really just looks just at audition on its own, particularly in a language context, and that works beautifully. But the auditory predicting the visual predicting the auditory never works for us.
Okay. Yeah, so I can probably show you the data. It wasn't really for us on the predictions, but more on the difference detection response. So, but anyway, we can discuss this. But the other point that you made, and it's in my mind as well, and I really relate to what you said, is the fact that the connectivity in babies when it comes to a task versus resting state is really not the same. And I was wondering if what do you think that we can how strong can we relate to the resting state connectivity studies in instance, like is it really reliable in the interpretation that we would make? I mean, I'm not arguing the results, of course, with a variety of strong association between brain regions, but can we really interpret this as connectivity? This basically would be my question.
I think that's a great question. I think that's one I'm probably going to have to think a bunch about. But I will say that I share that reservation that you have. So in some ways, resting state work has always been work that's been a bit complicated to interpret. What is it? And in different literatures and different hands, it's interpreted differently. And in the future work that I've sort of kind of pitched the idea is to get your feedback like on this, is that I was thinking of it as being a proxy for connectivity. And that's one way of thinking about resting state. But I share some reservation that that's really what it's encoding, because we also know, particularly in adults, that it also seems to be not exactly reflective of a task, but when someone's in resting state, it's not like there's nothing going on. And that's particularly true in babies, where they're usually what we're doing is they're either asleep or we're showing them some kind of movie, right? And so, yeah, I think that probably where we'll start is thinking of this interrelationship between task and resting state, in part because in NIRS, in part because we can record that in the same babies at the same visit. But ultimately, I think where I'd like to see this work going is something, if this is really a question about task-based functional engagement and anatomy, I think ultimately the question has to be answered through, say, something like NIRS, where you can really get really good task-based connectivity, in part because the babies can really engage in the task, right? Because they're out of the magnet. And MR, like we're got to have DTI, or we have to be looking at the real anatomy of the brain. And to me, that would be kind of like the next level that would resolve some of the ambiguity about the resting state.
Thank you so much.
Yeah.
I wonder if you know around what age do premature babies start to show top-down sensory prediction?
That's a great question. I would actually really love to do some adult studies. And that's one of the things that we're hoping to do in the lab this upcoming year. So one thing I will say is that the premature population and individuals who are born prematurely are very different across cohorts, across time, because the medical interventions that we've had available for them have changed so dramatically. And the medical complications they've had and the courses that they've had in the NICU are really incomparable between, say, an adult now who is born premature and a baby now who is born premature. So I just want to say that as a caveat, but I think it's possible that we will still find differences in these sorts of abilities in adults. And the reason why I think so is because if you look at language tasks, for example, in adults who are born prematurely, you can still find some impairments, even in, say, individuals who are at good like colleges and universities where a lot of our adult research happens. So they're clearly incredibly high functioning, right? But neurally they look different, and particularly in language tasks. So, which is where most of the focus on prematurity, such as I sort of read it, from a task perspective, focuses on language because there's so many deficits associated with language in premature babies, or at least that's the literature I'm most familiar with. So for example, in adults, this was a study at University of Pittsburgh, so a good university, a really good university, and these are contrasting students who were born prematurely and students who were not. You can show that there's almost a compensatory amount of connectivity in the brain when these individuals are listening to sentences when they're premature compared to if they were born full term. So I think one possibility with preterm babies is they don't have these sorts of abilities. It might be just something like prediction, it might be connectivity, or it might be something broader than that, certainly. I don't want to say prediction is the answer to everything with prematurity. It might also just be a cause of a reduction in connectivity. And I think what they're going to do over their life is they're going to compensate. I don't think they're going to catch up. I think that they're going to follow a different trajectory. And part of how we want to answer that question is by starting at adults and see what they look like. But that's all purely conjecture. We have no idea. We haven't done, for example, the infant studies at later ages. But I love that question. Thank you so much for answering or for asking that.
Est-ce qu'il y a d'autres questions? Il y a pas de questions. Can you hear, Lauren?
Yes, I can. Yeah.
Oh, perfect. Okay. So this might be kind of a naive question, but I understand that for these studies with really young infants, the parent is usually very close around, or the infant is not the parent. And so what I was wondering is there a possibility that the reaction of the parent to whatever stimuli is shown could influence the infant, or are they too young for that to occur?
Yeah, I think that's a great question. So let me also show you too, I think this might be what got you thinking about this. I have a picture of one of the families from the original task that we did. This is a family that I just really loved. We got permission to use the baby's name. Her name is Wilhelmina. She was a really wonderful participant in our task. And her father. So in NIRS studies, and I think my understanding EEG studies as well, not only are the parents very close, but they're often not, the task isn't concealed from them. So often in baby behavioral studies, we have the parent have sort of the task obfuscated from them in some way. So either they have something over their eyes or they're listening to music, so that we can't have any of those parental responses. So part of the reason we don't do that with something like NIRS is because the first reason is that we need the parent to help us keep the baby from ripping the cap off their head. So they often are really, really helpful to us to keep the baby's hands down. And the second is that by and large as a field, we don't think that this is what's happening. But I will admit that we've not tested it. So with the behavioral studies, the idea is that it would be possible, likely very early on in life, that if something changed and there was a subtle change in the parents, like they tensed up or they moved in some suggestive way, that the babies might have an increase in their attention and they might look slightly longer. And given that the dependent measure there is the length of time that they look, then we thought, there it really does make sense, I think, to like have the parent be unengaged with the task. Maybe the parent could also like shift one direction or another if the looking time is sort of differential right versus left. Here, the reason why I think that it's unlikely to have an effect is that we're finding effects, first of all, fairly locally in the brain. And I don't think a parent sort of general response of like, oh, there's something new is going to result in a specific response to, say, the occipital lobe. And particularly with our more graded findings of, for example, showing that the brain is really tracking prediction error over the entire experiment, if the parent was driving a big part of that, they would have to be responding in a really complex way over time. So it's not a naive question. It's a great question. It's one of those ones that comes up and is good to think about. And for me, it's kind of weighing pros and cons, because if you don't have good compliance with the task and you don't have babies not pulling off the sensors, we don't have anything at all. Do I know for sure the parents are not having an effect? I don't, but my suspicion, my very strong suspicion is this is just too sophisticated for the parents to be doing something to causing these effects themselves. But I do think babies at this age can be very sensitive to parental responses. And I will just add, sorry, this is a very long-winded answer, that's one of the reasons why we don't actually blind the parents or like, conceal the task from them. Because if the parents are relaxed and comfortable, the babies are actually so much more engaged with the task. And this is a big part of our experimental procedures, is to have the parents as comfortable as possible. It's an amazing question. I've sometimes thought about, particularly in behavioral studies, can we do a study where we actually try to get the parent to get their baby to respond, to really get to a real answer, at least in the case where I think they're most likely to have an effect. So we know, but there isn't anything out there.
And that's a good question, and I think that maybe some hyperscanning studies can help answering that too.
Yeah. Oh, that's such a great idea. I hadn't thought about from hyperscanning. That's wonderful. Yeah.
Maybe one last question. Is it okay with you, Lauren? We have a lot of questions that
I would be happy to keep taking questions. This is great. Yeah.
I was just wondering if you could explain the difference between NIRS and, for example, EEG, because I'm not very familiar with the technique. So help me like appreciate your talk even more.
You know what? I have to apologize. I debated, do I do my usual, like, this is what NIRS is? And I decided no, because I was like, you guys are going to be boring you. I'm sorry. That was a misjudgment on my part. So, so NIRS' closest analogy, or closest like analog, is fMRI, because both of those techniques are recording oxygenation in the cortex, by and large. And that's the sort of contrast that that people most often make. fNIRS, fMRI, fNIRS, fMRI, because of that physiological part of it. In the practice of NIRS, it is a lot more like EEG, because you're placing it on the scalp, and the ways in which you actually do the studies with the babies. So in terms of contrast with EEG, I would say that unlike EEG, NIRS is relatively insensitive to babies' motion and their eye movements, which is a real advantage, particularly for getting to see them and to see what their brain does when they're really active in these sorts of tasks. You can get motion effects with NIRS, of course, but there's easier ways to correct for them. And the motion effects, by and large, are very short in time, and the NIRS response, like fMRI, is quite long, over many seconds. So generally, we're not very sensitive to motion with NIRS, which is really helpful. But unlike EEG, it's very hard to get, you don't get temporal responses to particular periods of time. So for example, I think sort of illustrate this well. Sorry if I can find it. Oh, it's earlier. Here is this task, which was an EEG task, right? In an EEG task, and with the sort of MMN error responses that you typically look at with EEG in these sorts of paradigms, with EEG, what you would do is you look at the response to each of these individual shapes, and these shapes are also coming with sounds. But with NIRS, because it's like fMRI, we have to look for a much longer period of time. And so what we do is we're kind of grouping these shapes together over 10 seconds and looking at the response over all of them, because we've lost that temporal resolution here. But what we've gained is the ability to say something specific about a particular area of the brain. So that's how we're able to make inferences about the occipital lobe specifically versus the temporal lobe versus the frontal lobe. And it's not to say by saying that I don't think you can make spatial or sort of more functional inferences with EEG. And of course, you see that all the time with, for example, where in the time course it happens. Earlier components are thought to be perceptual, later components are not, are thought to be more cognitive. And in terms of sort of the scalp topography, as well as you can use source localization. But we kind of get it for free with NIRS because the channels are located over a specific part of the brain, and we're recording, by and large, that specific part of the brain. And for Anne and other folks there that use fNIRS, you know that all the ways in which I'm glossing over the details of surface vasculature and everything else, but that's the kind of how I think of the interrelationship between the two. It is a spatial temporal one, but they both, fNIRS gives you that additional robustness to motion in babies.
Donc, est-ce qu'il y a des questions urgentes ou des dernières questions? Parce que je vois le temps qui file, puis je ne veux pas trop prendre de temps à Lauren. So if it, so we will we'll close the session here. So thank you very, very much, Lauren, for the amazing talk. Really, it was super interesting, and for your time, for all the questions. And I will keep in touch in the next week. I'm going to contact you regarding this the system you asked about, the co-registration system.
Great, wonderful. Well, thank you so much. It really was a pleasure. I'm sorry I can't be there in person, but yes, thank you for the questions. They were wonderful, and it was a pleasure to share my work with you all.
Thank you.